Thank you for joining us today for the Benchling webinar, High-Throughput Molecular Biology from Your Living Room. My name is Sean Smith. I am a marketing manager here at Benchling, and I will be moderating today's event. Happy to be joined by two of my esteemed colleagues here. Firstly, Andrew Guo. Andrew is a product manager here at Benchling who is responsible for leading the development of the molecular biology tools. Andrew, how are you this morning?
Pretty good, Sean. Good to see everyone.
Would I be correct in saying that you're joining us from your kitchen this morning rather than the living room, looking at the background there?
Nope. Yeah, definitely in my kitchen today.
Great. And also joined by Damien Costello. Damien is an implementation manager who leads some of our most molecular biology intensive implementations and customer rollouts. Damien, you doing well this morning?
I am. I'm excited to get started here.
All right, excellent. Well, today's session was actually supposed to be part of a much larger in-person event, but obviously we've had to change the format, among other things, as a result of the COVID-19 pandemic. Damien, I noticed your hair's a bit longer than usual. I imagine you're probably looking forward to hairdressers reopening.
Yeah. I know that shelter in place has been lifted in a lot of places, but hair salons are one of the last to open, so I got it marked on my calendar.
Excellent. Well, look, I'm personally looking forward to a conversation today, not just about some of the new features that we've released in our molecular biology tools, but also, how scientists can use Benchling to stay productive, either while they're out of the lab or while we have limited time in the lab. So just before we get started, a quick housekeeping item. You can ask questions at any point during the webinar using the Q&A button on the panel below. And we'll get to them when we can. And without further ado, I'd like to hand over to Andrew to get the presentation started.
Great. Let me go ahead and share my screen. Great. So to give you all a quick outline of the agenda for today's webinar, I'll first start with a quick overview of Benchling in the molecular biology suite, and then pass it over to my colleague Damien, who's going to be walking through an antibody R&D use case that will highlight a few different tools within the molecular biology suite, both old and new. We'll then come back and provide some additional detail on some recent and brand new features that we've released, which will carry over into a roadmap overview of what's coming next. Lastly, we'll end today's session with discussion and Q&A. So as Sean mentioned, please feel free to ask questions below, and we'll try to answer some of those along the way. But rest assured, we'll have that longer Q&A session at the end of today's webinar. So please stick around if you have any questions. And with that, I will jump right in. So as many of you know, the world of life sciences R&D is complex and constantly changing. And that's really why we built Benchling, because we noticed this fundamental shift in how modern life sciences R&D orgs aim to work. We noticed that scientific workflows are evolving faster than ever, with groups looking to rapidly adapt to new scientific discoveries and broader industry macro trends, such as the recent shift of focus towards biologics. And two, as R&D workflows increase in complexity, life science teams have become increasingly specialized, with each phase of the innovation cycle requiring specific skills and expertise. And with the current coronavirus pandemic, companies have further accelerated moving to adopt a distributed team model. Regardless of how these teams are actually structured or distributed, they have to be able to continue to collaborate, share, and be able to coordinate processes effectively to drive progress on crucial company research goals. And finally, life science companies have to be able to build interconnected ecosystems of tools in order to rapidly incorporate new scientific innovations and technological advancements like CRISPR gene editing, lab automation, and genomics, as well as connect in data sources across their R&D departments to help drive intelligent, data-driven decisions. Unfortunately, the software to support this transformation hasn't necessarily kept up. Life science companies are being asked to rapidly integrate new discoveries and deliver more complex products faster than ever, and they are constantly running into artificial barriers of their legacy R&D platforms to do new things. Quite simply, software and digital infrastructure has not kept pace over the last decade, and many companies are left managing critical processes using pen and paper, spreadsheets, and homegrown technologies that are difficult and costly to maintain and don't necessarily meet the present or future needs of their R&D programs. We think every company and scientific team deserves a solution that will accelerate and support your innovations. So when you think about the ideal traits of any digital solution, there are really three core attributes to consider. One, the solution needs to be built for your complex science. It really has to be able to model all of the different molecules and biological entities that are specific to your organization's research needs. Two, the solution needs to be able to adapt to your processes. It has to be able to support all of the different processes and workflows that your team performs today and let you easily iterate and scale your operations while also being flexible enough to adapt as your R&D processes evolve over time. And three, it needs to enable key decision-making. It has to empower users with data at every single level, giving scientists additional insights about their experiments and team leaders additional data on their programs so that both can make the most informed scientific and operational decisions. And that's really why we built the Benchling Life Sciences R&D Cloud. Benchling's 7 applications span the R&D process from molecular design, experimental tracking, data acquisition, and process management. All 7 of these applications are built on top of a common unified data platform, meaning that data input in one application can easily flow through and be referenced in all other applications. This unified platform also houses an ultra-configurable data model that is able to map to each team's unique scientific and operational requirements, and ultimately makes it possible for us to query the system for extra intelligence using our Insights application. Benchling is built on a modern cloud infrastructure, fully managed by us and able to offer state-of-the-art security and control. And we know that for a lot of companies with a rich ecosystem of instruments, software applications, and additional data stores that their R&D teams depend on, we provide a full set of REST APIs that can help you connect in and out of all of these additional applications and instruments. Today, we'll be focusing on the molecular biology suite, though. And the molecular biology suite is really a whole suite of different modern tools that enable a variety of different molecular biology-related tasks, ranging from construct design and cloning to more specialized tools for CRISPR guide RNA design and antibody discovery. Damien is going to be walking through many of these tools in his demo shortly, so I won't go into too much detail on each of these. But I do want to highlight a few tools that we've added recently or made enhancements to that will be covered in the demo. Specifically, CODON optimization and back translation, some new antibody variable domain characterization tools, and a new Autofill translations feature, which lets you automatically link your DNA sequences to their registered amino acid equivalents. Again, we're constantly adding new features and functionality to our suite, and I'll highlight some more recent and upcoming additions at the end of our session. But today, our molecular biology tools are already being used by hundreds of life sciences companies that span a wide variety of research verticals, from gene editing to antibody discovery to your more historically large biopharma companies. And we're proud to work with all of these hundreds of leading companies. With that, I'll pass it over to Damien, who's going to be walking through a live demo of an antibody R&D use case now.
Wonderful. Thanks, Andrew. So I hop over and share my screen now. So what you're seeing here is the Benchling workspace, if you aren't already familiar with it. I have opened up here a notebook entry where I'll kind of be walking through an end-to-end kind of vector design process and documenting information along the way in my notebook. Today's use case that we're going to be running through is basically synthesizing a monoclonal antibody that targets the SARS-CoV spike antigen. So what you'll see here is an attached image which provides a rough overview of what we'll be doing. So we'll start with some files that I imported from Vector NTI. As you guys are probably familiar by now, Vector NTI is sunsetting their product on December 31st of 2020. So good as time as ever to bring your files in from there into Benchling. We'll codon optimize some of the chain encoding sequences. So the idea here is that we have monoclonal antibodies that were created in a human. We want to codon optimize them so that we can express them in an E. coli system. Along the way, we'll visualize a multitude of biochemical properties at the protein level. We'll assemble some expression vectors in bulk using Benchling's bulk assembly tools. And then we'll have Benchling analyze and autofill the translations on the DNA, creating a link then to the encoded protein sequence. We'll obviously then go to the bench, we'll synthesize those molecules at the bench, generate our mini preps, and have them sent to sequencing, only to, at the very end, visualize those sequencing results using Benchling's alignment tool. So with that said, we'll start with the first step here. It's to codon optimize our chains for expression in bacterial cell lines. In the interest of time, I've already codon optimized one of the sequences. So I'll walk you now through how to codon optimize the heavy chain DNA sequence. So clicking on this @mentioned chip links me into the molecular biology suite. One way to know you're in the molecular biology suite is to see the list of tools that we have here on the right-hand side. You'll see that on the left-hand side, I have a sequence view, and on the right-hand side, I have metadata. So this is a seamless integration between Benchling's molecular biology tools and our registry, which allows you to both visualize the sequence and capture critical information for that sequence at the same time. Before I codon optimize this sequence, I do have the capacity to create translations on it. For example, I can create a translation on the forward sequence here to generate the corresponding amino acid sequences. To take it a step further, I can actually do things like change individual amino acid sequences in the context of another organism. So in this case here, I can pull up a codon frequency chart that shows me, in the context of the E. coli organism, this is the most frequently used codon. So I can go through here, I can pick different codons if I choose, and I can do this at the individual amino acid level. Alternatively, I can actually analyze the translation as well, which will bring open a new tab called Biochemical Properties, which shows you things such as molecular weight, isoelectric point, extinction coefficient, and amino acid frequencies. Clicking into the sequence and dragging, you'll see that it actually updates real time with the corresponding amino acid frequencies based on the region that I've highlighted. So all of this is extremely useful if you want to look at protein properties at the level of the DNA, if you want to configure specific codons in the context of another organism. But as we mentioned, the first thing that we want to actually do here is codon optimize the heavy chain. The next thing I'm going to do here is I'm actually going to optimize this sequence for E. coli. So I'll highlight the sequence in bulk to do so. And you'll see that I have an option to then optimize my codons. In this case, I'm optimizing on the forward strand. You'll see that I have a few options here. I can pick the organism in which I would like to optimize my sequence for. I can toggle a variety of different GC content parameters, ensure that we have account for M-uridine depletion and avoiding hairpins. And you'll see that there's then 3 tabs in the codon optimization panel. One for protecting regions. In this case, Benchling has recognized 2 cut sites and assumes that I want to protect those regions. I actually don't. I can codon optimize and avoid the introduction of new restriction enzyme cut sites, or I can preserve cut sites that are already existing. So rather than having Benchling by default tell me which regions are protected, I'll actually specify here that I want to protect both my MBI and my NRU1 cut sites so that I can assemble them later down the line. I'll then click Preview Optimization, and before Benchling makes any changes to my DNA sequence, they'll give me this preview that you're seeing here. So at the top, it shows you the number of rare codons. We've actually depleted the rare codons to 1/3 of what we had before. you'll see that GC content and uridine content are relatively maintained based off of the parameters of the specifications that I had toggled earlier. And you'll see that we've made no changes to hairpin loops here. We'll also get an overview of kind of a before and after look at all of the codons that are being changed and the relative frequency in the original sequence versus in the context of E. coli. I can save these changes directly to my sequence, or I can save this as a new sequence and specify a location in my project structure where I would like to save those. You'll then see that Benchling creates for me a brand new codon optimized sequence here, presenting as well a translation on this sequence. Because this is a particular specific type of sample that I like to capture, I can toggle metadata such as this to capture important information against my sequence. Again, kind of an extension of Benchling's registry. And one of the tools that I actually can use here is this Autofill translations tool. So you'll see that when I do this, Benchling will actually create an additional translation for me now that it's recognized an amino acid sequence and directly create a link to this protein, such that when I click on this, I'm actually able to view additional information about this sequence now at the protein level. As Andrew had mentioned earlier, with our protein tools, with our antibody engineering tools, we can display very specific subsets of annotations, such as the CDR frameworks here. You'll notice that those biochemical properties that existed at the DNA level are present inherently at the protein level as well. And clicking on various CDR regions will update the biochemical properties for me real time. These CDR definitions can be toggled. You'll see that we have 3 different types of toggling that you can do. For CDR annotations, you can pick a specific definition. We provide Kabat, Chothia, IMGT, and North. We also provide for you a reference to the publication and the algorithm that we use to determine these or display these annotations. Below this, you'll notice 2 additional features. The first is antibody numbering. So I can actually zoom in on an amino acid sequence here if I want to toggle different things, such as the numbering or any post-translational modifications that I have. Hovering over these particular dots shows me, for example, the specific post-translational modification and where it exists relative to my CDR regions. Similar to the CDR annotation, I can actually toggle which specific liability sites that I want to see. In this case here, the ones that we provide out of the box range from N-link glycosylation to aspartate isomerization. Great. So we've codon optimized our sequences at this time. We've kind of viewed the biochemical properties for this sequence. The next step then is for us to bulk assemble our expression vectors. And so what we'll do is we'll use this pCry expression vector here. And you'll see that within this sequence, we have a LAC-Z promoter. So we'll be assembling 2 different sequences, a heavy chain and a light chain sequence, into the same backbone using Benchling's bulk assembly tool, the output of which will give us 2 unique plasmids, one containing the DNA for the heavy chain, one containing the DNA for the light chain. And we'll be using traditional restriction enzyme-based cloning methods to achieve this. So before I kind of get into the bulk assembly, similar to the codon optimization step, I'm curious to know what type of vector design tools you guys are currently using. And so we'll have another poll up here and just take a few more seconds to kind of toggle and see which plasmid assembly tools you guys are currently using.
Hey, Damien, just while we're waiting for that poll, I wanted to interrupt you with one of the questions we've had come through from Shuiwen Pan, which is, can you input sequence files in formats other than vector NTI?
Yes, you can. So Benchling supports a variety of different file formats. I don't currently have the list of them here. But anything ranging from an MA4 or PA4 archive file from Vector NTI to a multi-part FASTA, any GenBank files, any of the file formats that you might get from NCBI, etc., are accepted. And when we say accepted, what we mean here is that if you've previously taught or had annotations or characteristics displayed on those maps, we'll bring those annotations over along with it. Happy to provide a full list of the file formats that we currently support after this meeting today, if that's of interest. Okay, so it's time to assemble these. Like I mentioned, I'm going to use Benchling's bulk assembly tool. For those of you who are unfamiliar with this tool, you can think of it as kind of this combinatorial plasmid generator. In this case here, what we want to do is we want to use the pcry expression vector to clone in our two insert fragments. You'll see that there's 5 major steps for any given insert that you have. The first is to, of course, designate the sequence that you would like to use, as well as the particular strand of DNA that you would like to use. In this case, I want to use the forward method. With the bulk assembly tool, we offer 3 options. So the first is enzyme. This is obviously good for a use case if your fragments are flanked by restriction enzyme cut sites. Also, we support a variety of different Golden Gate-based restriction enzymes. So if you were doing Golden Gate assemblies, this would be the method you'd like to use. Homology is really good if you have some conserved sequence between your backbone and your insert fragment. For anyone doing Gibson assemblies, this is typically the method that we see you use the most. And finally, we have concatenation. So concatenation allows you to just piecemeal together any fragments independent of some technique that you would do at the bench, right? So it just simply allows you to create any combination of fragments that you would like to. As I mentioned, in this case, we're going to be doing restriction enzyme based. You see that we have a search functionality to be able to search for the particular restriction enzymes that we think are present on our map. What Benchling will then do is take the information that you've input here and present to you a brief display of it. You'll see that if I decide that I want to take the reverse strand, this will update real time for me. If I wanted to keep the smaller fragment to assemble with my insert fragments, I can do that as well. In this case, what I'm telling Benchling is take the larger piece of the backbone for me and insert in this white space here my 2 heavy chain, light chain DNA sequences. So for this next insert then, I can come back and I can pick my CODON optimized sequences. In this case here, I have a heavy chain and a light chain DNA sequence. You'll see that Benchling then remembers the restriction enzymes that I had selected on the previous step. And when I select these here, Benchling will go and do basically a summary report, show me which 5' and 3' sites are flanked by which enzymes, the length of that fragment, and basically do a validation to ensure that my overhangs are complementary, that it matches, etc. Again, as I mentioned, bulk assembly is a combinatorial vector design tool, which means that it will multiply all of the inserts that you decide to add into your assembly and create all of them. So in this case, it's 1 backbone times 2 inserts generates 2 unique plasmids. Of course, I can add multiple parts to this. If you're doing Gibson, you might have up to 15 fragments. So this big blue plus button here is how you would add on those fragments. So my last step here is to finalize it. Benchling, as you see, is warning me that I can't move forward with the assembly until I, 1, save these plasmids to a particular location. In this case here, I'm going to save it to my nested assembled plasmid folder. The next option that I have, which is, as you see, optional, is to add this to a work list. So you can think of a work list here as your favorites list. It allows you to take the plasmid maps, the fragments, the sequences that you work with most frequently in molecular biology, those sequences that might be distributed across a variety of different projects or subfolders, and combine them into this little favorites list to use in different parts of the product. So in this case here, I have an existing work list that I might want to add these to, in this case, my pcry vectors, or alternatively, I can create a new one. So I can say demo assembled plasmids, And this will create a new work list for me and move my assembled plasmids to it. Finally, and again, optionally, you'll see another integration between Benchling's molecular biology tools and the registry. I can define a particular schema for this. A schema in this case is a sample type, which again lets me capture standardized critical information for my monoclonal antibody vectors against the recently created sequences. You'll now see that Benchling gives me the okay to assemble these plasmids. And what you'll notice then is that Benchling actually opens or prompts me to open the assembled plasmids in an expanded view. And this is in an effort to do some, high throughput action against these sequences. So for example, one of the things that I can do here is I can click on this to select both of the unique plasmids that I've generated. I can come to this more dropdown and finally to analyze. You'll see then here that I have a variety of different options, one of which that we've already displayed to you before and that Andrew mentioned in his portion of the demo is this autofill translations. So rather than doing it at the individual amino acids or the individual plasmid sequence, you see that I can do it in bulk across all of the assembled plasmids that I have. This autofill translations then creates a translation on the DNA sequence, as you'll see in just a bit, and links me to the corresponding amino acid sequence so that I can essentially sanity check that the sequences that I just assembled actually encode the protein that I need it to encode. Another thing that I can do here in the spirit of collaboration is attach primers, right? So if I wanted to come into here and check to see if there were any primers that somebody had already designed for these pCry expression vectors, I can search across a variety of different places to find primers to attach to this. Rather than going and creating new primers for myself each and every time, I want Benchling to check and see if somebody else has created a sequencing primer. And if they have, I would prefer to use theirs and know that those have already been validated rather than creating my own and starting over again. You'll also notice that I did a quick refresh on the page. So after bringing in or creating a link between this pCry expression vector and the corresponding amino acid sequence, you'll see that we were also able to lift up some of the protein properties, such as the type of chain that it is, the molecular weight, the isoelectric point, etc., to the level of the plasmid map so that we can see that information without having to click out of the DNA. So let's look a little bit further here. Let's kind of dig in, right? So we click into one of these sequences here. I'll click on the home button to collapse my workspace and give myself a little bit more room. You'll see that I kind of have a plasmid-based view, which has its own set of display features that I can toggle on and off. And I also have my sequence view. I mentioned earlier that we were inserting these sequences downstream of the LAC-Z promoter. You'll see that that DNA sequence was inserted and a translation was created automatically for me. I can also click on the metadata, again, an extension of our registry, to see things like the chain type, molecular weight, and isoelectric point for the corresponding amino acid sequence. Again, I can click on this amino acid sequence to view biochemical properties, CDR annotations, definitions, numbering, post-translational modifications, chain type, etc. Okay, so I've attached sequencing primers in this case here. It looks like we didn't have any good candidates, which means that somebody else hasn't created primers for this plasmid map now, and I know that I have to create new ones. So I'm actually going to use Benchling's primer design tool to make these, specifically our primer wizard, to make sequencing primers to ensure that the prep that I'm eventually going to make at the bench is actually valid. Okay, so as I mentioned, the next step then is for me to go ahead and design some sequencing primers, right? So I know that, pretty much from the LAC-Z promoter down through the entire coding region of my chain, this is the entire region that I want to sequence, right? I need sequencing primers that cover this region. So rather than kind of curating my primers individually, one at a time, like I might do, what I'm actually going to do here is use Benchling's primer wizard to do this. So I'll use this selection here. You'll see that it automatically recognizes the region that I wanted. You'll see that for sequencing primers in particular, we have things like spacing, interval, lead, and accuracy definitions. Again, these are pulled from primer 3. So we're just using the same definitions that primer 3 uses for these parameters here. I'll click generate primers. And then what Benchling will do is go back and look at that and provide for me a variety of different primers that I can use. In this case, maybe I like all of them. You'll see selecting them toggles a display on the DNA level for me to reference quickly. And I'll choose to save these selected primers. I'm going to save them in my high-throughput molecular biology tools, and I'll probably want to save them in my sequencing results folder. So now, later down the line, if another scientist comes by and uses the pCRY expression vector or wants to use a prep that I've made inside the lab but doesn't know what primers to use to sequence, they can attach from existing and attach from my project folder to see if someone else has created primers in the past that have been validated. So a really useful way to kind of collaborate, especially around vector design. Great. So I generally know what sequencing primers I need to order now. I've designed them. I've assembled the theoretical vectors. Of course, then the next step would be for me to go to the bench, transform that in E. coli, and prep those DNA samples. In this case here, what we're showing you is a Benchling result table. So I had that Worklist, and I mentioned that Worklists integrate through the variety of different areas in the product. I can actually add those assembled plasmids that I assembled on the previous step and add them directly to this result table. Maybe I want to enter something like the concentration of the prep that I made. Doing so not only allows me to capture data against a particular sample in a structured manner, but again, fosters collaboration, right? So if somebody were to stumble across this expression vector later down the line, maybe they hadn't synthesized it, but they were interested in synthesizing it, we can actually see that in the results tab, we will end up having a result table that shows you the information that was specified for this particular vector. And it shows you who made it, what day it was made, what time it was made, whether that entry was sent for review, and then links you back to the notebook entry in case there was other information, cloning strategy information, a protocol that you use to synthesize these that another scientist could use, right? So, I would imagine making my primer mixture or my sequencing mixture. I take my preps, my primers, I throw them in a tube. I'd either sequence it in-house or send it out to a CRO to do sequencing. In this case, it's typically Sanger sequencing. And the last step that I would want to do then, once I receive my data, is to visualize the alignments. The last thing that I would want to do then is import my my AB1 files into Benchling. So you'll see here that in my sequencing results folder, I've imported some AB1 files. In the expanded view, I might want to create an alignment, right? So in this case here, I want to create an alignment for these three particular sequences. These are my AB1 files against one of the expression vectors that I assembled during a previous step. So you see I have my sequencing primers here. I have my AB1 files, my template. I select them. I come over to create DNA alignment. You'll see that Benchling then by default grabs those 4 sequences that I want to create an alignment for. You'll also then notice that I have an option to search for additional sequences that already exist in Benchling in the event that they are not nested in the same project folder. I can import additional files in the event that maybe I forgot to download or import one of the AB1 files that I got from my sequencing. Or for those wild childs out there, you can create your own bases and name the sequence on the fly to have as a reference sequence in your alignment. You'll also notice that by default, Benchling is choosing consensus alignment. This is because we're doing it through the kind of expand bulk action modal. It assumes that you're taking all of these sequences and wanting to make some sort of consensus from them. However, I can very easily just click on one of the particular sequences to designate it as the template. And now all of my kind of alignment results will be relative to the theoretical sequence that I assembled in bulk assembly. I'll choose to then create my alignment here. What Benchling will then do is actually open up my template map. So close this out here. You'll see that this is the same sequence map that we generated in the bulk assembly tool with our lag-Z promoter, our heavy chain here, the metadata corresponding to the DNA sequence with some biochemical properties. And now in this tab here is our alignment. So you'll see here then that everything that I got, this is our Benchling's alignment tool. I can compare my individual traces against my template map. In this case, I did a wonderful job with my assembly, of course, and I was able to sequence this without having any mismatches. One thing that I'll just call out is you can base call directly into this. So let's just say, for example, that this was a particular mutated region. You'll see that this then shows up as a mismatch both on our mini map below as well as in the AB1 sequence itself. And that's also visualized at the top by this score that you see here. You'll also notice that I have both annotations displayed as well as reading frames. So maybe I'm only curious about seeing one reading frame. In this case, I have 3. I can toggle the specific reading frames that I want to see in the upper right-hand corner. So in this case, I'm only interested in the first reading frame here. And we'll see that Benchling looks at the mismatch relative to the first reading frame and indicates that this particular mutation at the DNA level will cause a mutation at the amino acid level as well. And again, that's represented by the mismatch in the window that you see here below. So I've kind of gone end to end with this, right? I started with some sequences that I needed to codon optimize. I assembled them into my expression vector using Benchling's bulk assembly tools. I captured critical information against both the individual sequences as well as the assembled plasmid maps. This allows for folks to access my notebook entry or the results that I captured against the vector at the same time. I also went through and designed sequencing primers, which I saved into a project folder that other people can reference the next time that they want to design their primers. The last thing that I'll just call out here in the spirit of collaboration is once I'm done with this particular reaction or set of experiments, I can mention other scientists. In this case here, I might want Andrew Collins to take a look. This will actually send an email directly to Andrew Collins with a link to my notebook entry. So if I'm in the lab and Andrew's at home, he still has the opportunity to see the preps that I made, the concentrations that I got for them, the translations, the amino acid biochemical properties. He can also, get a link directly to my notebook and view my sequencing results as well. So with that said, I think we have about 15 minutes left. I think we will transition now back to some Q&A, and I will give the screen share back to Andrew so he can pull up his slides.
Great, thanks for that, Damien. Let me go ahead and share my screen again. So what I'm going to do now is that there's actually a number of questions in the Q&A box. Thanks so much, everyone who has asked questions. I've tried to answer some along the way and written out some responses, but some of them actually fit in really well with some of the roadmap discussions and recent features that we've launched. So I will try to cover those in this session now. The first thing I wanted to walk through is a few recent feature releases that we've put out in the last few months or so. Damien's kind of already talked about some of these items, such as bulk assembly, codon optimization, and Worklist integrations. But let me just step forward. For codon optimization, we've taken a open source algorithm called DNA Chisel and use that to power a lot of our codon optimization actions in the backend. There are a number of different parameters or things that it looks to optimize on, not just on the like removing rare codon usage. I will say that it the algorithm doesn't explicitly choose, there is no like prefixed or preferred codon that it picks every single time. It doesn't just use the most common codon to eliminate rare codon usage. Instead, it looks at the codon frequency table of the organism that you selected and tries to, I guess, play around with a few of different parameters, including trying to reduce the creation of different mRNA structures that could infringe on synthesis, such as the creation of hairpins, to also like optimizing for things like GC content or uridine depletion if those are set. And so it's really playing with all of these things in conjunction with some of the codon frequency information as well. I think Damien also showed an example of how you could protect certain restriction enzyme cut sites and protect certain regions to not be optimized in your action as well. And so these are all different parameters that kind of come into play when the algorithm is running. The one other question here is, can it perform silent mutations? The codon optimization will be performing these silent mutations underneath the hood when adjusting for those codon frequency usages. And so let's see. I'll just move forward now so that we can get through some of these slides. But as Damien also mentioned, that we've also added a concept of worklist in Benchling. Worklists are a great way to just move items around in Benchling, whether that's like saving items into a worklist and then using that worklist as input for another application or another tool within Benchling, or even just moving them around different tables and things like that. You can include any sort of entity container or batch into a worklist. And I really think of worklist as like a smarter kind of like copy to clipboard function, where you can actually like copy multiple entities to a worklist and then just specify that worklist somewhere else to be used, instead of having to pick all of the different discrete items across different folder directories and projects and things like that. And so we've already added certain integrations with Worklist, like being able to add primers that you've created directly to a Worklist, or being able to use Worklist as input and output of bulk assembly. But we're always adding additional ways to utilize Worklist throughout the platform. So that's going to be a piece of ongoing enhancements that we're going to take on. In terms of new feature releases, things that have come in the last month or so, the first set of enhancements are around improvements to our alignment tool. That's actually a hot topic / theme of work for us in this quarter. And so up until now, we've actually already delivered a few improvements to our DNA alignments tools, such that it can now take in larger alignments with more sequences and sequences of longer length. This is the first of many kind of performance-related improvements. We have some plans down the line also to integrate additional algorithms that would allow us to extend the capabilities of our DNA alignments tool to offer support for additional workflows, such as contig assembly, for example. I think there was a question there. And another set of alignments enhancements that we've made in the last month or so is around enhancing the PDF exports of alignments. So you'll see that we've actually added a lot more flexibility there in terms of exporting your alignments, both protein and DNA alignments. The other set of items I want to highlight are around the antibody engineering features that Damien mentioned, and also the autofill translations feature. We have very recently introduced an ability for Benchling to detect and annotate any sort of antibody-specific information on variable domains of your amino acid sequences or amino acid alignments. And this includes the ability to detect and add different antibody numbering schemes from Cabot to Chothia and IMGT, to additional ways to detect and annotate CDR and framework regions of your variable domains. And lastly, we've also added an ability to detect and surface certain post-translational modification sites or sequence liability sites on any protein sequence. So you'll see here an example of methionine oxidation. There was a question about bulk assembly with HCs and LCs to be considered together. So bulk assembly right now is primarily for assembling DNA constructs and not protein sequences. For
I can add some color there, though.
sure,
So for the actual assembly of, let's say, like a full length protein that has both a heavy and a light chain in it, rather than doing a two-step assembly with backbone insert one, having both fragments in that, you would do backbone insert one, insert two, where insert one is either your light chain or heavy chain. Insert two is your light chain or heavy chain, depending on what you pick for the first one. If there's a full-length protein that has an amino acid sequence with those two kind of concatenated together in Benchling, and Benchling during the auto translation run can read the codons from start to finish and create a matching amino acid sequence, that will work. But it does require some curation or a reference amino acid sequence to go off of. And the assembly strategy would look somewhat different.
great, thanks for that.
There were
There,
also some questions here just around moving away a little bit from the technical side and more towards the collaborative side. So one person asks, can you app mention or share to managers who may not use the notebook as frequently? Can managers view the registry, inventories, lab entries, etc.? The answer to that question is yes. We actually see that quite a lot, where there's a project manager or someone who's not going to be using the tools as extensively but still needs insight. Typically that's managed by a permission, such as, we give them read permission to the registry, to the inventory, to your notebooks. If they have an account in Benchling and you app mention them, that, they will get a link to that notebook entry. If they're not in Benchling at all, don't have any account, you can still generate a share link that provides them a link to the notebook entry outside of it. So it's a special type of link that you can generate. And then the other question, in kind of in the same vein, was, if somebody answers your questions or sees that you app mention them and has a reply, will you be able to see those answers? The answer to that question is again yes, assuming they go into the notebook entry and they leave some sort of comment, you'll be able to see that comment there. If they app mention you explicitly, you'll get an email, and then everything is captured in the version history of the notebook entry as well. So I could see a timestamp that says, Andrew left a comment at 10 a.m. on 6/12, and jump to that particular edit to see what they said. So those are some of the answers on some of the more collaborative based pieces. And then, Andrew,
awesome.
Sorry, I didn't mean to cut you off there, but
no worries. I just wanted to get through. There was some additional antibody related questions. Just wanted to get through those. So there's one question around, are antibody fragments supported by the CDR detection features? So currently, this is actually a planned enhancement that we're going to be rolling out in the next 2 weeks or so. Right now, our antibody detection limitations are, we will look for and detect a single variable domain and annotate a single variable domain per amino acid sequence. And this was mostly a factor of the open source tool that we use to build our tooling off of. The tool that we're using is called SAB-PRED, which is an antibody engineering toolkit provided by the Oxford Protein Informatics Group. We are looking to extend this functionality to be able to detect and annotate multiple antibody fragments on the same protein sequence. So in the situation that you had, detect and annotate multiple variable domains on a protein sequence. So in the situation that you had modeled an antibody fragment, or whether that's a Fab or like a single chain fragment, we would be able to detect and annotate multiple variable domains within the same protein sequence then. In terms of kappa and lambda annotations, we do differentiate between whether a light chain is a kappa or a lambda light chain. Great. And so in the interest of time, I'm going to move over to autofill translations because it's a little bit of additional content that I want to get to at the end about future roadmap. But for autofill translations, I think Damien quickly showed this. The main purpose that autofill translations serves is that it allows any user to automatically link any DNA entity to a registered amino acid entity within their registry. So in the situation that, for example, you are creating multiple antibody chains, you might have different DNA equivalents that all translate to that antibody chain, or you might have different like assembled plasmids that all have encode for that same antibody chain. And this autofill translations piece will actually allow you to allow Benchling to automatically scan through all 6 reading frames and see if any section of the DNA sequence actually can translate to any of the registered amino acids in your registry of a particular schema type. So you can basically direct autofill translations to automatically surface any linkages to amino acid sequences. So it doesn't rely on any sort of specific annotations necessarily.
Yep, and just to provide color, like in our demonstration, I had, an amino acid sequence that I was trying to create that was derived from a human. So I autofill the translation to see that amino acid sequence, and then after I codon optimize my sequence, I just run the translation, fill translations to kind of sanity check and confirm that I'm still linking to the same amino acid sequence. Because if it links me to something different, then obviously a codon was changed and it caused the mutation. So it's also really useful if you're like Andrew said, you're working with a variety of different DNA sequences that are encoding the same protein, just as like a sanity check to make sure you're curating things properly.
Great. And the last slide that I have here is around upcoming feature releases. We do have some additional features planned and currently being developed around enhancements to our alignments tool. And most of these I characterize as kind of like platform integration pieces. So making sure that a specific alignment within Benchling, you can generate a URL link to it so that you can embed a reference to a particular alignment in any other product within Benchling. So making sure that you can embed this link in Notebook or in Registry and things like that. And then we're also adding alignments to our global search tool, such that if you were looking for a particular alignment that you did in the past, you would be able to search for that alignment based on the alignment's name or any associated AB1 files that went into that alignment. Currently, all of this is kind of stacked or pegged to the template sequence that you align to. And so we just want to make it easier and more seamless to reference and link all alignments that you've done in the past. In addition to these alignments changes, we're also going to be extending our CODON optimization and back translation tool. We actually just recently added the ability to detect and avoid hairpin structures. And we're also going to be extending this to be able to function in bulk so that you can bulk codon optimize or bulk back translate a number of different amino acid or protein or DNA sequences in a single go. I will also mention that an additional parameter that we are looking to expose, but this might be a little bit further down the line, is around introducing those cut sites. Right now, we have an ability to preserve existing cut sites or make sure that we do not introduce certain new cut sites. The obvious next addition to that is being able to actually specify, I want to introduce this cut site at a particular area. And so that's also, and I believe it's a parameter that we can expose, and we just need to figure out where that fits into the roadmap here. So the last thing I want to mention is that we're also looking to add an additional way to create DNA sequences. Right now, registration tables within Notebook let you create registered entities really quickly and easily. And we want to make sure that for anyone who is going down that path to create registered DNA sequences or amino acid sequences, that they're able to immediately upload their sequence files into the registration table to basically produce these registered DNA or protein sequences in mass. So outside of the items that I've listed here that are coming soon, we are also going to be focusing on two different areas of work in the next 2 quarters or so that I want to mention. One is around certain enhancements to our bulk assembly tool, and this is initially going to include bringing over some aspects of our discrete assembly wizards into our bulk assembly tool. So the first example I'll bring out here is being able to bulk create Gibson primers from the bulk assembly tool. And then the other line of work that we're going to be diving into is around enabling the support of chemically modified sequences within Benchling. And so we're really looking to bridge the worlds of small and large molecule development and make sure that any of our users who are currently working with modified RNAs, DNAs, or peptides are able to accurately model, design, and attribute results to those entities within Benchling. So that's going to be a pretty large theme of work for us for the next few quarters, but I just wanted to call that out as something to look forward to in the future. And I know that we are about at time now, and I tried to get through as many questions as possible throughout the roadmap section, but were there any that came in the end that you can try to answer at the end? Or Sean, are we at time now?
Yeah, I think we've got about 1 minute left. So just before we choose a final question, I just wanted to put in a plug for our next event. So if you could just move to the next slide there, Andrew. For anyone that is interested in a more general overview of the Benchling platform, you can tune in on the 8th of July. We're going to have an event by the called Elevating Virtual R&D Collaboration While Out of the Lab. And that's going to include a live demo, an overview of Benchling, as well as a customer panel discussion with that will include some folks from Maverick Therapeutics and Verve Therapeutics. Really excited about that. And we are bang on time. So I guess one question that I wanted to finish on, and before I do, for those questions that we haven't got to, I will make sure that we respond to you individually. But just to finish it off, I'd be really interested to hear your thoughts on what tools or features you're seeing most important for our customers that are currently partially remote.
Yeah, on the implementation side, I'd say the two that are most heavily used, app mentioning is a big one. App mentioning users, app mentioning other samples, other notebook entries. That's one that folks adopt very early on when they migrate into Benchling. From the molecular biology side, auto annotate and and attach primers from the bulk expand view is another frequently used one. People will often build out custom feature libraries that they collaborate on together to tag their plasmid maps or to have Benchling go back and search for particular regions so that they can quickly identify components of plasmid maps that they might not have generated themselves or might have generated outside of Benchling and without the ability to annotate. And then attach primers is another very commonly used one, just given the fact that people are sequencing all the time. They don't want to have to recreate a primer every single time they go. And the chances are that if they're working on, some commonly used vectors, they have a predefined set of sequencing primers that they would want to pull from and see who's designed it, when it was designed, do they have inventory of it, rather than starting over each time. So those are the big ones that I see. And the one that I see on the horizon is just work lists, especially as we start to build out permissions around them, the ability to share work lists, things like that, to allow people to kind of grab a predefined set of samples and move to the next step.
Awesome. Thanks so much, Damien. Out of respect for everyone's time, we'll wrap it up now. Thanks so much for joining. And you can see other events that are coming up at www.benchling.com/events. Thanks, guys.
Thanks, everyone.